So my job today in the next 45 minutes is to talk something about how to manipulate the gut microbiome. You heard a brilliant talk by Dr. Uday Ghoshal, who actually set the stage for my talk. So how many of you believe that God is omnipresent? Okay, I'm not sure, but microbes definitely are present everywhere. Okay, so if we see the National Geographic TV channel or news or the magazine, you'll see microbes in the high temperature volcanic vents. It's seen under the Antarctic glacier. It's actually seen in spacecraft coming back from space. So it's microbes available, present even in space. That has been speculative. So from where do these microbes come from? So initially it was thought that the amniotic fluid is a sterile space. But now there are studies which have shown that there are microbes in the amniotic fluid, and the baby could actually acquire microbes even in utero. So do you know this man? Anyone? Okay. So this is Udzi. Actually, what happened a few years back, in the Swiss Alps, some mountaineers found this mummy. So they thought some mountaineer had died falling from the cliff, but later on, by doing sequencing and carbon dating, it was found that it was a mummy from 4,000-year-old. So that was a very good opportunity to see the gut microbiome in that mummy. And they also studied 8,000-old coprolites. Coprolites are fossils of stools of ancient people. Right. So when they looked at the microbes in all these patients, the people, like I always tend to say patients because I always see patients. Okay, so it was seen that there was lots of similarity in the microbes in the modern man and in the ancient man. And if you see, there is lots of similarity, lots of overlap, which means this has been conserved. The gut microbiome has been conserved since long time back. So what do the microbiome do in the gut? So it has lots of metabolic function. It has metabolic, it causes lots of drug metabolism, gut barrier integrity, and immunomodulation. And when we talk about gut microbial manipulation or protection, we deal mostly with the immunomodulatory properties. Okay, so how does antimicrobial protection happen? Why the gut in the intestine do not infect us? Why didn't it come into the systemic circulation? So the mechanisms are a little bit different in a large intestine and small intestine. And you had already heard from Dr. Ghoshal about the intestinal mucosa and the mucus. So the large intestine has a very thick mucus layer, around 100 to 150 micron thickness, and there are two layers actually, the outer layer and the inner layer. The outer layer is a very dynamic layer where you could see lots of mucin, lots of resident bacteria and nutrients and free fatty acids. And the inner mucus is a very stable layer. So the microbes cannot traverse this thick mucus from the lumen into the intestinal lumen, into the intestine and go into the systemic circulation. But in the small intestine, the mucosal layer is very fragmented and very thin, it's broken. So here some other mechanism has to play a role. And you already heard about MAMS, membrane molecular these patterns, and then PAMS. So when these organisms, the pathogens, are invading the intestinal lumen, all this gets activated. The pattern recognition molecules gets activated, and they start secreting antimicrobial peptides from the Paneth cell, and they start killing this organism. So that's how the pathogens cannot enter the host circulation, and they do not cause system. So basically, a low-grade inflammation continues in the intestinal lumen, which protects the individuals from pathogenic organisms. And the other thing which is important is it produces lots of anti-inflammatory molecules. It activates the intestinal dendritic cells and activate the Tregs, the T helper cells and Tregs, which are protective for the host, to the host or the people. It also has a role in gut microbial integrity by maintaining the tight junctions. It inhibits apoptosis of the enterocytes, and it reduces the endotoxemia. And there are several studies, I'm not putting the references, but these are the basic mechanism by which the gut integrity is maintained. Now, when is the microbiome altered, or when are the microbes altered? So this is the list of functions that I had just briefly touched upon. And these are a list of diseases now where lots of association has been seen. And again, you had heard from Dr. Ghoshal a long list of GI diseases where the microbes are altered. Now there are evidence that even in diabetes, atherosclerosis, obesity, autism, there are changes in the gut microbiome. So we call it dysbiosis. And mind you, these are all association studies. Very few studies have actually shown that these changes actually cause the disease. So still the story of the chicken and the egg is going on here. So we really don't know which comes first, which comes later. But in some diseases like ulcerative colitis and irritable bowel syndrome, hepatic cancer, what we already heard, in those diseases, the gut microbes actually have been shown to be shown to cause the pathophysiology or pathogenesis of the disease. So what determines the gut microbiota? There are several factors, right from birth to old age, different factors like the mode of delivery, the early feed. Is it a breastfed baby or a formula fed? Is it a cesarean baby or is it a normal vaginal delivery? So all these things are the earliest factors that determine the gut host microbiome. And as you grow, the diet, the meat-based diet or the vegan-based diet, or if you're taking processed feed, so all these things, the geography, antibiotic use, biodiversity, and of course host genetics. So these factors will affect how the microbiome is shaped over the entire lifetime. Now, when we talk about microbiome, and I had just showed that there are lots of factors that could affect the microbiome or shape the microbiome. So it's logical that since the world is a very diverse place, in the West, the genes are different, the diet are different. In India, the diet are different, genes are different, the diversity is different. So is there a difference of the Indian microbiome compared to the West? And there has been few studies, and one study was from this institute led by Mojibur. We were also collaborating in that, where 15 tribal populations were taken from all over India, 4 from Telangana. And what was found is there was significant similarity in the entire microbiome in the tribal population of the species at the genus level. And there are a few core bacteria like Prevotella faecalibacterium, Eubacterium. These were the overall core microbiota, but there were some differences in Manipur and Sikkim, probably because of their altitude and the dietary factors. A similar study was published from Pune, where they compared the microbiota in rural and urban people. And if you see here, the rural samples were clustering close together, which means that the rural lifestyle, which is more of vegetarian, close to nature, they had a more conserved organism. And even in that study, Prevotella was predominant. And is it different from the West, or is it same from the West? The Indian microbiota is definitely different from the West. If you see, compared to the American gut microbiota, there was significant difference in the relative abundances of some of the organisms. And even in the tribal study, when the microbiota of different tribes all over the world was compared, the Indian microbes was closely similar to the Mongolian tribes. So it shows that Indian microbes are different, both in urban, rural, and tribal population. And therefore, whenever we talk about manipulation of the gut microbiota, it has to be customized to our population. But the problem is we do not have specific studies, except for a couple of studies from Dr. Ghoshal's group from All India Institute and PGI Chandigarh, where gut microbial manipulation of Indian diseases were shown to be benefited. So why do you do microbial manipulation? So now I had already said these microbes are associated with diseases. The Indian microbes are different from the West. So when there is dysbiosis, logically it comes to the clinician's mind that why don't we modify the bacteria and try to make it into a good bacteria so that the patients get benefited from the disease. So that's the basic objective. The main goal is to give back their normal microbial function that I had already mentioned. And there are several old methods. There are several new methods coming up, and that is what I'm going to discuss today in the next 35 minutes or so. So how many of you have taken probiotics so far? No one? Okay. And why did you take it? Diarrhea or just gastric? Okay. So how do we define probiotics? Probiotics are defined as live microorganisms. So when you administer it the adequate amount, they confer a health benefit on the host. And then along with probiotic, the other concept is prebiotic, because the gut organisms require some source of nutrition. And many of our food cannot be digested by the intestinal enzymes. So these bacteria can digest those and produce short chain fatty acids. They get their nutrition and give a health benefit to the intestinal cells. So these are called prebiotics. So basically, these are fermented ingredients that result in specific change in the composition of the organisms. Now, when we talk about probiotics, one which you get in the market, it consists of only a very small number of organisms. So lactobacillus species, bifidobacteria, E. coli specific, this Nissel 1917 strain, Bacillus again very specifically Clostridium specifically Butyricum, and Saccharomyces boulardii. But if you see, there are so many species of organisms in the intestine. So the first question that I would ask, is this enough to cause a change in the gut microbiota, to make the gut microbiota healthy? So if you see the market, you'll see like men's probiotic, women's probiotic. They have even love bugs probiotic. Maybe it will start increasing love more. For kids are different. All of you have seen about Yakult, right? You have taken Yakult. So are these basically marketing gimmicks, or do they really have a therapeutic benefit? Right. So let's see something into the history. The concept of probiotic actually dates back to 1899, when a scientist called Henry Teaser isolated bifidobacteria. But Metnikov gets the credit of the first for the father of probiotic, because he was the one to develop the concept of using this as a probiotic. Right. Then in 1917, Alfred Nissel isolated the E. coli Nissel strain, and 1935, someone from Japan called Minoru Shirota isolated the Lactobacillus casei. And if you look at the publication history, right from 1954 to 2018, so there has been a sharp rise of this publication from 1997. And this morning, this year, when I looked at PubMed, there were at least 255 publications in just 1 month on probiotics. So that shows that probiotics are increasing interest of the physicians, scientists, but at the same time, many of these studies are funded by the pharmaceuticals. So, and pharmaceutical funded studies are published easy. There are many mechanisms, I won't go into those. So that is kind of a good thing, but also kind of a concern. So how much true data that we are delivering to the literature, okay?